gave a seminar here sometime within the past year or so. And he's also been made a little bit famous lately for trying to educate some of our politicians on climate change.
Not me, Jeff.
He then went on to get a postdoc at MBL, and last year was promoted to an assistant research scientist. And so I guess you're going to tell us a little bit about what you've been doing up there?
Well, yeah, some of that, and some stuff you probably haven't heard about that I did down here. So, yeah. All right, thanks, Chris. So thanks everybody for hanging out late on a Friday. So I appreciate y'all coming out and paying attention. Hopefully I won't be too boring. So, so I'm an ecosystem scientist, but I work with animals, and so and for the most part, animals are largely not really thought about much in ecosystem science. And for the longest time, they're just sort of considered response variables. Something happens in the ecosystem, and then it sort of drives what animals do. Animals really weren't capable of moving a lot of energy, moving a lot of nutrients, or changing the way an ecosystem was structured. But everyone sort of knows examples where this isn't the case, right? That they're not just response variables. There's great migrations that occur in Africa that move, where big herds of animals move up and down the continent and take with them all the productivity they had fed down in the valley and move it up and down. And everybody knows the classic example of the salmon that spends most of its life out in the ocean and comes back upstream. And it's a subsidy from the ocean to terrestrial habitats. And the reason that these things are sort of difficult to quantify and look at is we really didn't have the tools, but now we have the tools and the capability with things like stable isotopes and these new metrics and looking at specific elements and things like compound-specific isotope analysis and using some innovative methods and things. We can actually track these and quantify the roles animals play in moving nutrients and energy around ecosystems. So just to give you a couple examples, this was a one that I became aware of where that fish secrete as part of their salt pump to extract water from marine fish are always drinking. To extract the water from their guts, they actually use this calcium ion exchange pump, and what they form these calcite crystals in their guts and excrete them. And a really clever Scottish guy figured out did a calculation, and this accounts for about 14% of all the carbonate in coastal ocean sediments, which is quite a lot. And another example recently from the literature was there's a couple of them actually that just came out where whales via their feces, so by consuming phytoplankton, and then all whales do poop on the surface. So by bringing they consume krill, and then they bring their poop back up to the top of the water, and that keeps these essential nutrients in the water column and allows for the production of phytoplankton to keep occurring. So animals, and this accounts for quite a bit, as much as 10 million times the concentration of iron in the Southern Ocean in a whale plume than the surrounding water. So these things can be they can be fairly important for maintaining primary productivity. So the one I want to talk about was the one that I did here in Florida that maybe I didn't do a really good job of publicizing. Maybe I should have done a better one. Was this flux by fin idea from the Big Bend seagrass beds out to the coastal ocean in the Gulf of Mexico. So right outside the door here, or just a little north, there's lots and lots of seagrass. And in that seagrass, there's lots of charismatic fauna and not so charismatic fauna. But one thing there's definitely a lot of all the time are these pinfish. And so, you're asking me how many pinfish. Well, actually, the guy sitting up here in the front of the room counted them, or you didn't count all of them. And so it turns out there's about 1.5 billion individuals produced annually in the Big Bend seagrass. And that's about 10 to 15 million kilograms of biomass, and 90% of that biomass is produced in 1 year. So that's quite a lot of biomass from these seagrass beds. And this is about 3,000 square kilometers of seagrass. It's one of the only contiguous ones left in the Western Hemisphere. But it's still sort of temperate in this area, right? So it gets cold. Not quite cold enough to snow, but it gets cold. And these fish egress, they leave the seagrass. And so you have this export of productivity from the seagrass beds, and when they migrate offshore to spawn. And when they get there, they run into these guys, not the fishermen, the fish. So these are gag grouper, and so these are the best fishermen in the Gulf of Mexico, in my opinion. And so gag grouper are very important economically to the state of Florida. They help support the 5.2 billion. I think it's even more than that now. $5.2 million, and that's the biggest one y'all have ever seen because he was 5 pounds bigger than the world record. Gag grouper. That's me standing behind Dink, who caught him. Yeah, that was a big fish. And so they also eat pinfish like popcorn. And so this link, this subsidy, could be an important thing that helps support this higher trophic level predator out here in the oligotrophic Gulf of Mexico, where there's not a lot of primary productivity. And so to sort of tease this apart, we decided to use stable isotopes. So this is just the vanilla version of carbon. So it has 6 neutrons, 6 protons, and 6 electrons, right? Carbon 12. And there is, if you add a neutron, you get carbon 13. And it looks, tastes, acts, and smells the same, except for it's just a little bit heavier, which causes all the reactions with carbon 13 to react just a little slower, and makes the bonds a little harder to make and break. And so we can use these subtle differences to track the isotopes because you literally are what you eat, right? The atoms you consume in your mouth get rearranged, and you're composed of the things that you've eaten. So we can use these stable isotope values to track energy through food webs. So for this talk today, I'm going to be talking about, you'll see carbon and nitrogen values. So carbon and sulfur are source tracers. So they look pretty much like the primary production source that laid them down. And nitrogen is a trophic tracer, so it fractionates. Every time it's handed off from one consumer to the next, it gets a little heavier, and so each trophic level you go up, you get a little heavier in nitrogen. So you'll have a few box plots of that later. So here's our Big Bend seagrass beds, and this when we sampled in the Big Bend, we used data from Chris Dowling's work, and we worked with people from the FWC and National Marine Fisheries Service in Panama City who were surveying these shallow water reefs, and then we caught gag grouper out here in deep water, and then we also caught gag grouper out in deep water, so all completely removed. None of them were inside the shallow, less than 30 meters. Or I think most of them were actually caught in 50 meters. And in the deep water reefs, we caught all the potential prey items on those reefs as well. Oh, look, and so here was this is the Madison-Swanson Marine Protected Area, and then these are all the spots where we caught the grouper. And so when we plot this, we have sulfur on our y-axis, we have carbon on the x-axis. And so here's our deep water prey species, our shallow water prey species, and here's our gag grouper that plot over here above seagrass. And when you do a mixing model, you find that the seagrass bed contribute to gag that are about more than 100 kilometers away from the seagrass, contribute around 18.5 to 25% of the tissue in that grouper came from the Big Bend seagrass beds. And so 18 to 25% of these big upper trophic level critters is composed of secondary productivity that was produced more than 100 kilometers away. So what I wanted to do, so my original thinking, this is what's great about science and doing science is, how does this stack up? How does this compare to other sources of primary productivity in the Gulf of Mexico? And then, are they overutilized? So is the pinfish, because it's 25%, so my thinking was there's no way that pinfish are 25% of all the productivity. So this subsidy was overrepresented in the grouper's tissues, right? So they were using it more because it was a prey item. So what I started to do was I actually compared the sources of nitrogen in the Gulf of Mexico, assuming that the Gulf of Mexico is oligotrophic and nitrogen limited. So using nitrogen as my currency, I assumed all these fluxes, 100% of the flux was turned into animals and moved up 3 trophic levels to be equivalent to pinfish biomass. And all of these estimates were done in total nitrogen, and none of it we didn't consider. The recycled nitrogen is not considered because it's already in the box. And we don't know a lot about upwelling. Maybe someone here can tell me if there's a lot of upwelling in the Gulf, but we ignored upwelling as not a source as not an extra source of nitrogen in our estimates. And when you do that, so we use the Apalachicola River and the Suwannee River as riverine fluxes. We looked at atmospheric deposition, fixation of nitrogen by trichodesmium, and submarine groundwater discharge. And so these are the basins. So this is the Apalachicola River Basin. So this drains here. This is the Suwannee River Basin. And then we had a 70,000 square kilometer box that we used for doing our aerial estimates of nitrogen fixation by trichodesmium and aerial deposition. And then here's the Big Bend seagrass beds. So you can see this is nitrogen that comes from a lot of, fairly substantial areas relative to these seagrass beds. And so the source of nitrogen was assumed to go from nitrogen to the primary producer, secondary consumer to the tertiary consumer, and then to our groupers. And then the pinfish was set at trophic level 3. So there's 3 trophic transfers here, and then this pinfish can be directly consumed by gag. And when you do that, when you look at it, it actually turns out that pinfish biomass, even if you took all those other sources of nitrogen and turned every single bit of it into animal, that pinfish are approximately 25% or 23% of the total available productivity if 100% of all the sources that we used was turned into fish. So the animals are a huge flux of material out to the Gulf of Mexico. That is compared in the oligotroph, and obviously 100% of this stuff does not get turned into fish biomass. So these fish are a critical link supporting productivity probably above and beyond what the ecosystem just in the Gulf of Mexico could support at such a high trophic level. And if you think about it in terms of losing seagrass structure, it's almost a 1-to-1 loss. You would lose literally that export, and you would lose that grouper meat each year, each time you lose an acre of seagrass. So that's pretty cool. And it kind of blew me away because I figured there'd be no way that it would be actually 25% of the total available. So it worked out, and it's kind of cool. So these are examples of animals facilitating movements of nitrogen and movements of energy, and actually controlling, in some circumstances, ecosystem level processes. So they aren't just response variables. Animals matter in ecosystem science, and they can be important. We need to understand what they're doing to help understand how ecosystems work. And so, moving beyond that now, what are the factors that control these animal-mediated processes, and how will they respond? How animals respond and help shape the way future ecosystems look as climate change happens? And we go on. So I left sunny Florida for Massachusetts.
Is that how you get to work?
Well, I'm exaggerating. We use snowmobiles.
And that was in 1960.
Right. So now I'm going to take you all the way up to actually, it was beautiful up there now, but it can be not so beautiful. To the Plum Island estuary. So I work in the Plum Island long-term ecosystem research site. It's fabulous. If you get an opportunity to work with these places, they have really great long-term data sets. I highly recommend it. If you want to come hang out with me in the marsh and get muddy, I highly recommend it. It's beautiful, and we have lots and lots of long-term data. And I'm going to show you some of that long-term data and how it's really great at looking at these kind of questions. So it offers you a unique opportunity to sort of long-term, long-scale look at process-based stuff, abiotic driving biotic processes, and you can study these in these coastal ecosystems where obviously there's a lot of important ecosystem services that happen in the coastal area and in coastal areas, and understanding those processes are important to maintaining those services. So fish facilitate energy production by concentrating and transporting nutrients across the sea. So the movement of the organisms is important for transporting nutrients and nutrient energy. And this process, so this is sort of where I work from in my research right now. This process is enhanced by the geomorphic features in the marsh. So having rivulets, having marsh interfaces, having areas of high and low marsh, and transferring that in areas where you can produce things and they get shifted are important at controlling the productivity that we see. And so this idea is not super new. It's not super new. And so this is sort of Ron Kanive's trophic relay. So animals that are smaller, they gather energy from very productive areas, and then as they get larger, they move, and then they connect with other predators that consume them, and so you have this sort of handing off of energy. And so the new component, the way I'm thinking about it now, is that there's these processes of excretion that help stimulate primary productivity, and these processes of consumption that help remove the productivity from one place to the next, and that's facilitated at different geomorphic scales by these features. So you have these small, shallow places that provide structure and habitat for small critters. They gather their energy, and then they're moved to these larger areas. And these areas where you have discontinuities and connections of habitat are hot spots for transferring energy via the movement of these animals. And so I'm going to talk about one thing that we can do to mess this all up. And so this is the tide experiment. And if you haven't heard about it, you should check it out because it's very, very cool. It's an ecosystem level experiment. It's been going on for a decade. We just got it refunded for 3 more years. We add 70 micromolar nitrate to the incoming tide water in these tidal creeks, and to simulate what anthropogenic nutrient pollution does to the marsh. And we monitor everything. We do this at So we have a long-term enrichment that's been going on for over a decade. We have a shorter-term enrichment that we started back in 2009, and we have control creeks. Each one of these creek sheds, as we like to call them, are 6,000 hectares, and we monitor the plants, the microbes, the fish, everything, all ecosystem processes, nitrogen, the sediments, all of it. And so we have this really awesome long-term data sets, and we can ask these sort of big questions. And so, in looking at how animals responded to this nutrient press, there's sort of a couple ways that you can have. There's sort of this strong interacting top-down view of the world where you have these very strong connections and you get these very strong movements. So if you stimulated more plant productivity, that would make more snails, and then the snails would get eaten by the crabs, and so you have this very strong trophic interaction. Or you can have these very reticulated food webs where the energy is sort of displaced because, say you produce more primary production, that'll end up in your primary consumer, but then you have all these reticulations where the energy can get spun off and lost, and so you can have in this instance, you might expect sort of a weaker response to the nutrient press. So here you'd expect a strong response and an upward, and then here it would just be dissipated and you might not see anything. So when we look at our food web, so the fish that we're going to focus on today is the wily mummy chog. Now mind you, I went from studying fish that were like this to studying fish that are like this. So it was a little, to call him a top predator, which he is in the marsh, is sort of funny, but he is. So he's in my marsh system. This would be my apex predator. They consume these little shrimps and primarily these small invertebrates that I don't know the names of, but David Johnson does, luckily. And which are so he's our primary predator, and then these are the secondary consumers. And so, but we have this very sort of looks like a reticulated kind of food web, right? There's lots of places this energy could go. There's dead ends in the food web. There's loops in this food web. So we would expect to see sort of not these sort of strong interactions. So the community sort of observations that were the responses of the community. One, we added nitrogen, so we made more algae, right? That's sort of obvious. We add more algae. So we have more algae, and we also got lots more of these small invertebrates. So then we have a lot more of these. So here's the reference creek. And this is biomass of these snails. So we got lots of these grazing snails. And at first, in the first 6 years of the nutrient enrichment, we had this very strong response by the nectin, by the fish. So we had this very large increase. So this is the biomass. This is the peak biomass of the enriched divided by the reference, right? So when we started the experiment, the reference creek was actually less productive in terms of nectin than the experimental creek, and it went up to about 2.5 times the biomass. So this implies that there was these strong linkages, forcing energy up the food web to the top predator. Now, if the theory holds, what would happen is that once this energy comes into equilibrium with a new rate of production, right, you would expect this to top out, right, in some new equilibrium with the productivity being put in the bottom, right? But of course that didn't happen. So we had since 2009, it's done nothing but go down, and this year it's even lower. So we've lost fish productivity. So now the energy is going somewhere else, but we're still producing the algae. There's still lots of snails. There's still lots of other things, but we're not producing the fish. And so that brings me to right. So now we're wondering why this is sort of completely counter to the bottom-up strong linkages idea, and it's completely counter to what we think should have happened. And so I'm neglecting to tell you that the one other major response, and this is from the paper in Nature in 2012, was we saw that these creeks, and there's a dog for scale, these creeks, one of the major responses we had from these creek systems was that the low marsh, so this is Spartina patens, which is a high marsh. It's sort of hard to describe because in southern places, they're low. Everything's very low hung, and like where Juncus, would be this is where Juncus would be, but there's a 9-foot tidal prism here, so this is very high peat bench. And the low marsh, this Spartina alterniflora, this bright green area, is Spartina alterniflora. It's a tall form. It grows to be about a meter. And so what that in this fringing habitat right here on the creek edge, what happened was this plant grew taller, but because we were adding nitrogen to the system, it grew less roots. So it was putting down less biomass into the ground. And it was and by adding an electron acceptor, by adding the nitrate, we stimulated microbial respiration of the sediment. So we had less stabilizing roots, and we had more microbial respiration of the sediment, and the creek banks started to collapse. So the creek would literally crack and cave, and it's amazing. There are these cracks right along the edge of the high marsh where the high marsh meets the low marsh that are 100 meters long. It's incredible. And so, all right, look at that. And so now we have this sort of very interrupted habitat so instead of having these nice benches where you have lots of access for fish and things, now you have you have a lot of, it's, what's the word I'm looking for? It's patchy. It's more fragmented, right, thank you. It's much more fragmented habitat, so you don't have these nice connections. And it turns out that these accessing these areas in the high marsh is actually very, very important for the growth of these fish. So what you have here is this is Spartina contribution to the fish, and this is the number of days that they get access to the high marsh. So when you have more access to the high marsh, there's a greater contribution of this Spartina habitat to their tissues, and then the greater the flooding frequency, the greater the growth. And so what we think now is that we may be limiting the access to these areas, and we're keeping the fish in high concentrations where there's just algae. Well, Jimmy, if there's lots of algae, you stimulate lots of algae, why aren't the fish could just switch and eat algae, right? They're omnivorous. So I'd had this little experiment, and I love this picture. So we did this experiment this summer with one of my REUs where we had fish that we fed them algae only, grown out of the water from the creeks, detritus only, so just Spartina detritus, and then we fed them crickets, so things that they would actually get on the high marsh. And so as you can see, the fish didn't die, so we put all of the fish in the tanks we had them there for 10 weeks, we put them all in there at 15 millimeters. And so the fish did not die. They can subsist on algae, but they certainly do not thrive. They need the protein. So access to the high marsh protein is super critical for producing fish. And so now my working hypothesis is that this slumping and caving of the creek bank decouples the high marsh habitat from the creek bank habitat, and you lose the movement of energy across the high marsh down into the creeks where the fish are. And so you're decoupling the habitats, and you're severing the link, and you lose